So thank you for the invitation. Let me start the session. So this is my talk, but I wanted to tell you a little bit about the session in general. So I'm going to be focusing on schizophrenia and neuroinflammation. We're all neuroimagers, or we're using neuroimaging in our research. So my talk is going to be about how neuroimaging can inform neuroinflammation hypothesis in schizophrenia. Then we are going to expand on the topic of proteomics and using peripheral biomarkers in combination with neuroimaging to study schizophrenia, but also other disorders like aging, like depression, and finally like eating disorders. So back to my presentation. My name is Marek Kubicki. I'm professor of psychiatry at Harvard Medical School. I'm also a trained radiologist and MR physicist, so I've always been interested in using neuroimaging in neuropsychiatric disorders, and ever since I started my lab in 2002, 2003, I've been using neuroimaging, and I was trying to use it to simply differentiate schizophrenia from healthy controls and finding the methods that would work best, the regions that they would show group differences. And that was the beginning, and we've kept doing it. We've kept increasing resolution, improving our sequences, and at some point we started asking ourselves, yes, but okay, there are differences. Whatever neuroimaging methodology you're using, you are able to show differences between groups. But what does it mean biologically? What are the mechanisms of action? What peripheral and central mechanisms these neuroimaging findings are related to with regard to schizophrenia? So today I'm going to be talking about a particular hypothesis of schizophrenia that involves neuroimaging. I will tell you how we got to the point where we started testing this hypothesis and what neuroimaging findings are related to and how we're going about looking at the mechanisms of neuroinflammation in schizophrenia. I have control of this? All right. I have nothing to disclose. This is more or less what my talk is going to be about. I'm going to try to tell you about where the hypothesis of schizophrenia is coming from, what data and what evidence is it based on. Then I'm going to talk about all neuroimaging methods that have been applied over the years to study neuroinflammation. Then I'm going to focus on what we are doing in our lab and the method that we are using, which is free water. I will tell you about the studies and the results we obtained using free water in schizophrenia. And then I will change the gears and I will talk about this one particular hypothesis and model that we're testing with free water. It's called MMP, or the protein that we link to free water is called MMP9. And I will tell you about schizophrenia and MMP9, and then our imaging and MMP9. All right, so going back to the beginning, the schizophrenia and neuroinflammation. It's the idea that has been put forward a long time ago in 40s and 50s. People were talking about inflamed brain and how this could be related to neuroinflammation, but because there were no good, reliable measures to detect neuroinflammation in the brain, this hypothesis was dormant for a long time, and in early 2000s it got revived, and it got revived in a big way. People started looking at CSF and pro-inflammatory and anti-inflammatory cytokines, measuring them and finding that they are pro-inflammatory cytokines that are abundant in CSF in people with schizophrenia. There are large, like this study from 2009 is a large population study showing that subjects who are on anti-inflammatory medication, not necessarily related to schizophrenia, are less frequently developing schizophrenia than the general population that is not on anti-inflammatory medication. There are also studies showing that, well, at least initial studies involving this one particular PET radioligand have been demonstrating that the activated microglia, which this PET ligand is supposed to bind to, have been upregulated in schizophrenia. Unfortunately, this methodology or this ligand has been disproved to be working in schizophrenia, so there are still no PET ligands that would show this process. However, in postmortem brains, individual studies as well as meta-analysis is showing increased density of activated microglia. There's also evidence about increased leukocyte T and B densities and suggestion that there might be a inflammatory subtype of schizophrenia. And there are even percentage numbers that are being assigned to this. Some people believe it's between 30 and 40% of schizophrenia population that might have this inflammatory subtype. But, I mean, one of the reasons we became interested in schizophrenia, or one of the reasons the schizophrenia hypothesis is so appealing is because of studies like this. Like, these are two examples of studies and results that almost never happen in our research world. So on the left, it's interleukin-1, which is pro-inflammatory cytokine, and it's upregulated in schizophrenia to the degree that you almost never see in research. And on the right side is CC4, and it's genetic study that shows that this particular gene among all the genes studied in schizophrenia is really upregulated, and it's involved in synaptic pruning and inflammation. Since we cannot really measure neuroinflammation in the brain without looking at postmortem brains, and those are from brains of people who are usually dying after chronically suffering from schizophrenia, and we cannot look at either dynamics of this process or starting point of this process. From the pathway which inflammation usually takes, we can select a couple of different consequences of neuroinflammation and maybe focus on them and maybe measure them. Glial activation increases the blood-brain barrier permeability, which increases the water content, creates edema. So either measuring glial activation or blood-brain barrier permeability or degree of edema in the brain could maybe give us some insight into how inflammation is involved in this disease. And there's a list of available right now imaging measures that could be applied and could be indirectly measure those consequences of neuroinflammation. One, like I said, is a TSPO PET ligand, which binds to activated microglia in the brain and is supposed to show the upregulation of glial cells. There are two measures of blood-brain barrier permeability, perfusion imaging and dynamic contrast enhancement. The dynamic contrast enhancement has been used in schizophrenia. It has been shown it's the injection of the contrast that is being used in brain tumors, for example. And people inject this contrast and then look at the signal from the brain and find that if there is permeability or there's blood-brain barrier disruption, there's contrast going into the brain and signal changes from before to after contrast. And there's at least one study that is showing that 35 to 40% of schizophrenics have this effect, this leakage of the blood-brain barrier. MRS is another measure that has been applied to study schizophrenia. It's, again, non-direct measurement of inflammation, but it measures some metabolites that might be involved in inflammatory pathway, like choline, myoinositol, and glutathione, which is antioxidant. And finally, diffusion MRI, which measures water content, which measures edema, which might be related to or consequence of neuroinflammation. We, in our lab, love diffusion imaging. That's majority of the imaging work that we do. So I'm going to be talking about diffusion imaging and how diffusion imaging can measure inflammation, basically ignoring all those other measures or other methods. So for those who don't know what diffusion is, it's a method that can tag the water molecules in the brain and can detect how well and how freely they are moving around in brain parenchyma. And they are moving depending obviously on the barriers that they are hitting, those water molecules, and this water is restricted either in all directions or one direction, like if you imagine, sorry, that's not what I wanted to do. So you can imagine that these are axons that are running parallel to each other. They're surrounded by myelin sheaths, and there's water in between them. And you can imagine that this water can move along the axons much more free than perpendicular to the axons. So in those areas with, we call it high anisotropy, in those areas with very tightly packed myelin surrounded axons, the water movement is going to be a little bit more ellipsoid than in the areas of CSF or in the areas of gray matter where water either cannot move at all because there are neurons and processes restricting this diffusion in all directions, or in CSF it moves in all directions freely and equally. It's also isotropic diffusion, right? So you can imagine that this anisotropic diffusion, it's hard to, the shape of this anisotropic diffusion can change if anything in the brain changes, right? If the axonal density increases, this anisotropy increases. If the myelin decreases, there's more space in between the axons, and diffusion and water can move freely perpendicular to the axons. So anisotropy decreases, fiber crossing decreases anisotropy, glial scars decrease anisotropy, and edema obviously increases anisotropy because there's more water. So like you can see, this diffusion is very sensitive to everything that happens in the brain, very non-specific to any particular biological marker. In order to make it a little bit more specific to edema, we developed this method called free water imaging. One of the members of my lab, Ofer Pasternak, came up with this model that instead of looking at diffusion and kind of having one model to characterize water diffusion all over the brain, it now contains two different compartments. One is extracellular and one is tissue related. He developed it because in a brain tumor, for example, brain tumor is surrounded by edema, and there are fiber bundles, fiber tracts that are traveling through edema, and they are sometimes infiltrated and damaged by the tumor, sometimes they are not, sometimes they're just surrounded by water. And this method was to get rid of edema and visualize, like, this is These are tracts. So on the far right side are the fiber tracts, the way they are visualized before the free water is being applied. And when free water is extracted, removed from the image, you can see that around the tumor, all of a sudden, we start seeing those fibers, those red fibers that are there. So obviously, it gives the good information to neurosurgeons who are operating on the tumor that there are still intact axons, and they should not be removing this tissue that is just it's just edema, and when edema goes away, the fibers will still be intact and working. So we took this free water, and we it's probably not a good order. So we took this method and we applied it to schizophrenia, because we thought that if there's a hypothesis about inflammation and there's a method that can measure inflammation, or edema at least, we might be able to detect it in brains of schizophrenics. And this was the first paper that we published in 2011, and we indeed found that people with first episode schizophrenia have very enlarged this extracellular compartment, free water compartment. So there is some edema going on, and it was all over the brain. And then in chronic schizophrenia, the same method applied to chronic schizophrenia showed only very limited increase of free water only in the frontal lobes. It's still there, but it's much more limited than in chronic schizophrenia. This red color is this other compartment, which is related to tissue, but it's not related to inflammation. This is a replication study on a completely different independent sample, and we show the same effect also. First episode schizophrenia shows much more free water than healthy controls. And we even, based on those preliminary data, we got a grant where we were allowed to harmonize 13 different data sets together, and we're showing the distribution of FA along the schizophrenia, like age trajectory and duration of illness, and we were able to show that free water is much more increased. This is tricky. Much more increased in very young people, and with the duration of illness that is very short. And free water differences, even though they're still existing, they go down with duration of illness and age. And then I should be showing this slide, which is, we were wondering, obviously, about free water sensitivity or specificity to neuroinflammation. So we performed the animal experiment, where this is the mouse model of maternal immune activation. So you inject the poly-IC to mothers, and then if in the offsprings it creates neuroinflammation, and then you can measure it with free water. And this is what we did, and we found that indeed in those animals the free water is increased in frontal cortex. So having all those findings about free water, we started obviously thinking about the mechanism on which we could test this. And the idea was to be able to manipulate the free water signal, at least maybe in animal models, but maybe also in patients with schizophrenia, to give them something that would either block and decrease or increase the levels of free water. And we were presenting our free water findings at one of the Schizophrenia Research Society meetings, And we came across this investigators, or we started talking to these investigators from the University of Lausanne, who had this beautiful model of MMP9, which was oxidative stress and neuroinflammation related, and it was showing all the phenotypes associated with schizophrenia, this model. So MMP9s are the extracellular acting zinc-dependent proteases that are expressed both in prefrontal cortex, in hippocampus, in choroid plexus. They can be measured in the brain, in the blood, in the periphery, and peripheral and CSF concentrations of MMP9 are correlated with brain concentrations, so it goes in and out of the blood-brain barrier. They are involved in extracellular matrix, so extracellular water, extracellular matrix, we thought there would be a relationship. They're involved in neuronal growth, plasticity, synaptic elimination, and what was the most important, in inflammation. They promote they promote increasing the blood-brain permeability and for the inflammation to go from periphery, from the blood into the brain. There are already studies looking at MMP9 in schizophrenia patients. MMP9 is upregulated in the blood, it's upregulated in the CSF, its upregulation in peripheral blood can be reduced, downregulated by the antipsychotic treatment. And there is association between higher MMP9 and increased risk for cognitive impairments in schizophrenia. MMP9 is also very well documented to be related to cognitive deficits, not only in schizophrenia, but in other neuropsychiatric diseases. And the idea of the Lausanne group and the collaboration that we started was to test this hypothesis, which is basically stating that there's a vicious cycle where it starts from redox, the oxidative stress dysregulation during brain development, and then it leads to MMP9 activation, which then initiates the inflammatory cascade. The upregulation of MMP9 in the brain leads to increased peripheral MMP9 levels, which then turn to increase blood-brain barrier permeability, influx of leukocytes into the brain parenchyma, and then the increase of extracellular free water. So basically, once the oxidative stress starts during development, that it never ends, and it culminates at the very early stages of schizophrenia onset. So with the schizophrenia onset, this vicious cycle is the most active. And we have applied, we started working with this group, and we started looking at the peripheral MMP9 and brain imaging. There's not that many data sets that have both brain imaging or diffusion imaging and blood stored and available for analysis. We found a couple of samples, and in one sample we showed correlation within hippocampus volume and MMP9 peripheral concentrations. And in another paper that was just published by Johanna, who's going to talk next, we found both upregulation of MMP9 activity in periphery was much higher in schizophrenia than in controls, and it was correlated with free water in the brain in patients, but not in controls. So this is very promising and gets us going. Obviously, what we so far know that free water diffusion can be a potential biomarker of brain inflammatory processes in early and acute phases of schizophrenia, that MMP9 peripheral upregulation seems to reflect oxidative stress-initiated inflammatory response, that MMP9 upregulation seems to explain blood-brain barrier disruptions, free water increase, and could be a target for novel therapeutic approaches. And that what we don't know is the dynamics of this process, whether it's upregulated and then downregulates. We don't know whether all people that have increased MMP9 levels in the blood have this blood-brain barrier disruptions and have this inflammatory processes happening in the brain, because we still don't know, because the free water is not a brain measure of inflammation. It just indirectly measures consequences of potential consequences of inflammation. So one of the things that we're doing, other than validating and increasing our sample and looking at MMP9 at different stages of schizophrenia, we're also working actively to develop a PET ligand that would go through blood-brain barrier and attach to MMP9 in the brain and show us brain distribution of MMP9 and activation and potential up-regulation of MMP9 if it is true in patients with schizophrenia. So with that, this is the group that is actively working on all those, or helped me with all those findings and working on all those projects. Boston team, our collaborators outside, and funding. And Johanna obviously is going to talk more about proteomics and interaction between imaging and blood measures of proteins, and then Lauren is going to talk about the same thing, except for not in schizophrenia or aging, but in eating disorders. And that's all. If you have any questions, I'm happy to answer either now or after the talk. All right.